Hi, my name is Craig and welcome to this channel, which is about image analysis, image processing and imaging in science. I thought I would begin a new series and make some life science videos. Basically short videos that introduced a piece of science, something that was topical. And of course, most topical thing at the moment, perhaps SARS-CoV-2 and Covid19. Now, as a cardiovascular scientist, I was fascinated to discover that ACE2 was involved. Now, in the cardiovascular world, ACE2 is fairly commonly known. And you may not have heard of ACE2, perhaps that's why you're watching this video. So what I wanted to do is to make a fairly short movie that explains what ACE2 is and why it's important in Covid-19. May give us some therapeutic targets, who knows. Well, I hope you enjoy this, and I may make some more of this type of video if it seems popular. Thank you. The lungs are a prime target of the virus, and here we see a cross-section of a small airway in the lungs. The epithelial cells that line the inside of the airways and alveoli express the ACE2 protein on their cell surface, and it's that protein that the SARS-CoV-2 virus can bind to. Similar cells exist on the inner surface of our blood vessels and therefore also offer a target for the virus. I'm going to sketch out a component of the renin-angiotensin-aldosterone system, and for that I need a liver. It's mainly the liver that produces angiotensinogen, a protein comprised of 485 amino acids. This is quickly cleaved to 452 amino acids and becomes the substrate for renin. So for this I need a kidney, as it's the juxtaglomerular cells of the kidney that release renin in response to various physiological conditions. Renin catalyses the formation of the 10 amino acid or decapeptide angiotensin 1. Now, angiotensin 1 is not very active or potent in the vascular system, but angiotensin 1 can be cleaved to an 8 amino octopeptide called angiotensin 2, and this is an extremely potent vasoconstrictor. And that cleavage is catalyzed by angiotensin converting enzyme, or ACE. Since angiotensin 2 is such a potent vasoconstrictor, it will raise blood pressure. Angiotensin 2 can be further cleaved to angiotensin 1 to 7 by ACE2. And angiotensin 1 to 7 is actually a vasodilator, so it can reduce blood pressure. So the balance of activity of ACE and ACE2 is extremely important in regulating blood pressure, and it's ACE2 that is the target for the SARS-CoV-2 virus. This 3D model of the SARS-CoV-2 virus was developed by virologists at the University of Glasgow and by medical animator Annabelle Slater. It can be found on the Glasgow Life Science Sketchfab page. The most striking features are the spike proteins that cover the surface. It's these spike proteins that can bind to ACE2 protein on the surface of epithelial and endothelial cells. The virus also has a lipid bilayer that we can see in more detail in the next model. So here's that lipid bilayer that is the surface coating of the virus. This is the inner part of the virus, and this is a single spike protein. It's worth mentioning that soap can destroy that membrane, and that's why it's so important to wash your hands to have a better chance of killing the virus. But it's this spike protein that's the real menace, because by binding to ACE2, it can gain entry into our cells. So blocking the binding of that spike protein to ACE2 could be an effective treatment. Here we see what is called a cartoon model of protein structure, and it tells us about the way in which the amino acids are arranged and how the protein is folded. We see ACE2 on the top with the virus spike attached. The space-fill model gives a better idea of what that ACE2 and spike complex looks like, and you can see how nicely they fit together in this animation. This is a model of a blood vessel, which can also be found on the Glasgow Life Science Sketchfab page. In the model, we see the external elastic lamina in white, which gives structure to the vessel. We see a nerve running along the surface of the vessel in red. And in green, we see the collagen and elastin structural matrix. There are also some fat cells here in yellow on the surface. Now in Sketchfab, with the model loaded, we can drag the model round to look at the inside surface. Now this inside surface here would normally be completely covered in these purple endothelial cells, and it's on these endothelial cells that we would normally find the ACE2 protein expressed at the cell surface. We can zoom in for a closer look, which isn't very smooth, using a PC mouse and on Sketchfab, and we can tilt the model to have a better view of the endothelial cells. And so here we see the ACE2 protein. We see the virus attacking the ACE2 protein, and now we see the view from inside the endothelial cell with the ACE2 protein on the membrane facing out of the cell. But when the virus binds to ACE2, the whole complex is internalized, and this enables the virus to hijack the cellular mechanism and replicate itself. These new virus particles can then exit the cell to the bloodstream, and once these new particles are out of the cell and in the bloodstream, they can go off in search of other cells to infect. Well, I hope you enjoyed that. If you liked it, remember to press like, and if you'd like me to make some more of this type of life science video, then leave me some comments and don't forget to subscribe. See you next time.